is Sumarendra Mohanty on behalf of Nanoscope Therapeutics.
Good morning, and thanks to Sibor for the opportunity to speak. I'm Samar Mohanty, co-founder, president of Nanoscope. I'm an inventor and have financial interest in the company. Today, I'm excited to share our groundbreaking work in bringing the light of hope for patients with severe vision loss. Next slide, please. We are leveraging 5 critical knowledge areas to facilitate development of fast disease-agnostic in vivo gene therapy to restore vision. These represent a paradigm shift from the traditional gene-specific approach. By recognizing the shared pathology across different diseases, we are paving the way for mutation-agnostic gene therapy. At the same time, we have been leveraging heterogeneous real-world data to establish clinical meaningfulness of our endpoints. We are also exploring how safety and efficacy may be influenced by pre-existing conditions across indications. Throughout the development, transgene expression data have been fully integrated to inform our decisions. Ultimately, our goal is to build a disease-agnostic platform for neurodegenerative eye condition, allowing us to apply safety and efficacy insights across multiple indications. Next slide. Let me paint the picture of why this approach is so desperately needed. The challenge is that the IRDs represent the most complex therapeutic areas in medicine today. We are dealing with over 8,000 mutations in more than 250 genes that have been associated with IRDs. And remarkably, 1/3 of these mutations remain unknown. Current gene therapies are limited to patients with precise genetic diagnosis. This leaves the vast majority of patients with no treatment options, especially those with very rare genotypes. These approaches also become ineffective when the target cell, the photoreceptors, is not present in the advanced stage of the disease. Next slide. Next slide, please. Here is where the disease-agnostic approach like ours can solve these challenges. Despite the incredible genetic diversity underlying IRDs, there is commonality in the disease progression pathway. Whether we are dealing with RP, Stargardt, choroideremia, the end result is same: progressive degeneration of photoreceptor leading to vision loss. This shared final pathway of cell death represents our therapeutic opportunity. By targeting the bipolar cells that are preserved in these conditions and delivering to them the tools to restore vision. Next slide, please. To do this, MCO10 therapy combines the best from gene and cell therapy approach. Instead of trying to fix the broken genes or replace the dead photoreceptor, we are reprogramming the surviving retinal infrastructure. MCO10 works by delivering proprietary engineered opsin to non-light sensing surviving bipolar neurons, transforming these cells into photoreceptor. A single intravitreal injection reprograms the retinal circuitry, allowing patients to regain vision regardless of which gene or disease caused the blindness. Next slide. The second critical area where we are breaking new ground is establishing clinical meaningfulness through real-world data analysis for over 70,000 RP patients in the AO registry, which shows that natural progression results in a decline of 1.5 letters per year. For the first time in a randomized trial in RP patients, we showed 0.3 logMAR improvement equivalent to 15 letters. That means a vision that is lost over 10 years is gained on average. This durable improvement has been observed over 3 years and expected to continue. Next slide. We are also trailblazing in understanding how pre-existing conditions affect treatment outcome. Most patients have neutralizing antibodies against AAV from childhood exposure. Yet, remarkably, our studies indicate that MCO10 therapy improves visual acuity regardless of baseline NAV values. We have treated patients across dozens of mutations and with therapeutic doses with no reported SAE. Importantly, the patients had evidence of improvement irrespective of gene mutation. And looking holistically across endpoints, those who did not improve in visual acuity had evidence of improvement in mobility or self-discrimination. Next slide. The fourth area where we are making advancements is in integrating transgene expression data throughout the product development. We are using red fluorescence imaging to track gene delivery to target cells and measure the retention across cells, animals, and humans. This comprehensive approach allows us to objectively determine the potency and optimize dosing across development cycle. Next slide. Finally, Our disease agnostic platform allows us to leverage safety and efficacy data across multiple neurodegenerative conditions of the eye. Our RP clinical results show statistically significant visual acuity improvement as compared to controls. When looking across endpoint, most patients has evidence of improvement in 2 out of 3 key efficacy assessments, demonstrating the broad functional impact. Importantly, all subjects had evidence of improvement in either of the 3 key assays, and the majority of MCO subjects improved in visual acuity. To summarize, our disease agnostic approach represents a paradigm shift in treating retinal degenerative disease with a universal platform, and the implications are far beyond retinal degenerative disease. Thank you for your attention.
Great, thank you so much.